Hi, everyone. From the outside, many bacterial cells can look fairly simple. They are small, they lack membrane-bound organelles, and under the microscope, they may appear to be little more than tiny rods or spheres. But inside the cell, an enormous amount of activity is taking place. Cells must store genetic information, build proteins, generate energy, respond to environmental change, and coordinate thousands of chemical reactions at the same time. Even prokaryotic cells, which are often described as simple, are highly organized and remarkably efficient. In this lecture, we'll explore the major internal structures of prokaryotic cells and how those structures support life at the cellular level. By the end of this module, you should be able to describe the major internal structures of prokaryotic cells and their functions, explain how internal organization supports metabolism, protein synthesis, and survival in prokaryotes. Most of the inside of a prokaryotic cell is made up of the cytoplasm. The cytoplasm is a gel-like internal environment surrounded by the plasma membrane. This is where many of the cell's chemical reactions occur. The cytoplasm contains enzymes, nutrients, ions, ribosomes, and many other molecules needed for metabolism and survival. As you look at the TEM micrograph on this slide, notice that the inside of the cell appears darker than the surrounding background. That darker region is the cytoplasm, packed with proteins, ribosomes, DNA, and other cellular components. Although simple diagrams sometimes make the inside of a cell look empty, the cytoplasm is actually extremely crowded and highly active. Thousands of chemical reactions can occur simultaneously inside a single microbial cell. One major challenge for every cell is storing and using genetic information. Unlike eukaryotic cells, prokaryotes do not have a membrane-bound nucleus. Instead, their DNA is located in a region called the nucleoid. As you look at the micrograph on this slide, notice the lighter region inside the cell labeled as the nucleoid. That lighter appearance reflects the area where the bacterial chromosome is concentrated. In most bacteria, the chromosome consists of a single circular DNA molecule. This DNA contains the instructions needed to build cellular structures, regulate metabolism, and respond to environmental conditions. Even though prokaryotes lack a nucleus, their genetic material is still highly organized inside the cell. In addition to their main chromosome, many bacteria also contain plasmids. Plasmids are small, circular DNA molecules that exist separately from the chromosome. These molecules often carry genes that provide useful traits. For example, plasmids may contain genes for antibiotic resistance, toxin production, or the ability to metabolize unusual compounds. One important feature of plasmids is that they can sometimes move between bacterial cells. This transfer of genetic information allows bacteria to rapidly spread useful adaptations through populations. Cells survive by building proteins. Proteins function as enzymes, structural components, transport molecules, and signaling molecules. Protein synthesis occurs at ribosomes. Prokaryotic ribosomes are called 70S ribosomes, which are smaller than the ribosomes found in eukaryotic cells, which are 80S ribosomes. Despite their smaller size, ribosomes are incredibly important because they translate genetic information into functional proteins. Because bacterial ribosomes differ from eukaryotic ribosomes, they are also common targets for antibiotics. Some antibiotics work by disrupting bacterial protein synthesis while having small effects on human cells. Cells also need ways to store important resources for later use. Many prokaryotes contain storage granules inside the cytoplasm. These granules may store carbon compounds, phosphates, sulfur, or other nutrients. As you can see in these various micrographs, storage granules can appear in many different forms, depending on the organism and the material being stored. Some cells contain large, bright inclusion bodies, while others contain many smaller granules distributed throughout the cytoplasm. Storage allows microbes to survive changing environmental conditions where nutrients may not always be available. Some aquatic microorganisms also contain gas vacuoles. These structures help regulate buoyancy, allowing cells to position themselves at favorable depths in the water column for light or nutrient availability. These storage structures help microbes survive fluctuating environments. For a long time, scientists thought prokaryotic cells lacked internal organization, but modern research has shown that bacteria are much more organized than we once believed. Many bacteria contain structural proteins that function similarly to components of the eukaryotic cytoskeleton. These proteins help maintain cell shape, assist with cell division, and help organize materials inside the cell. Some bacterial proteins even help position chromosomes and cellular structures within the cell. So, although prokaryotes do not contain membrane bound organelles, they still possess sophisticated internal organization. In this lecture, we explored several major internal structures found in prokaryotic cells. The cytoplasm provides an environment for metabolism. DNA stores genetic information. Ribosomes build proteins. Storage structures help cells survive fluctuating conditions. Together, these structures allow even very small cells to carry out the complex processes required for life. The organization of the cell directly supports its function. And although prokaryotic cells are structurally simpler than eukaryotic cells, they are still highly efficient and remarkably sophisticated biological systems. With that, thank you for attending this lecture, and we'll see you at the next one.